Hello everyone, and welcome to day 3 of the Cell Therapy Manufacturing and Gene Therapy Digital Week. I'm your host and this week's producer, Sarah Linton, and I'll shortly be handing over to our presenter for today's first session, Professor Mark Lowdell. We have 4 webinars left to bring you over the next 2 days, and your registration will give you access to all the sessions. Janneke Mullenberg's presentation will be following immediately after this, and you can also find on-demand recordings of the 2 sessions from day 1 on the homepage. And now, for today's first session with Professor Mark Lowdell, ISCT Vice President of Europe and the Professor of Cell and Tissue Therapy at UCL. He's speaking on preclinical and phase 1 trial considerations to prevent failure at marketing authorization approval. The PowerPoint presentation will last roughly 45 minutes and will be followed by a short Q&A with Mark. If you have any questions, whether during the presentation or after, please do type them into the box provided below at any point, and we'll try and get through as many as possible at the end of the presentation. Finally, if you have any technical issues, please click the red Live Support button and someone will be able to help you with any problems. So with that, I'll pass over to Mark.
Hello, I'm Professor Mark Lowdell, and today I'm going to talk to you about the important considerations that you need to think about when planning your preclinical and your phase 1 trials to lead to a successful marketing authorization for your ATMP. There are a number of disclosures that I should make, and they're highlighted in slide 2. But most importantly is to note that what I'm going to talk about today are my personal opinions and not those of the International Society of Cell Therapy or my employer. So moving on to slide 3, at the end of last year, we still face the scenario that more than 90% of advanced therapy medicinal products are being developed by investigators and not by drug companies. And about 60% of clinical trials are also investigator-led through Europe and the US. The remainder of the 40% are either from academic spin-out companies or spin-off companies or biopharma and pharmaceutical companies, and we'll talk about those in detail later on. But there's an argument that there is a paucity of licensed ATMPs worldwide, and one might argue that that's actually due to the maturity of the field. And I think if you think about the history of commercialization of monoclonal antibodies, you'll find that the commercialization rate of ATMPs is probably better even than historical experience with monoclonal antibodies. Kohler and Milstein published their first paper on the production of monoclonal antibodies in 1975, and it wasn't until 1986 that the first FDA licensed monoclonal antibody, OKT3, was made available. It took a further 8 years for the second antibody to get a license. And by 1998, 23 years after Kohler and Milstein published their first paper, there were still only 8 monoclonal antibodies in clinical use. Compare that with the 5 cell and gene therapy products that are already licensed by the FDA within 13 years of US legislation allowing such things. I think that the rate of commercialization of ATMPs actually is perfectly acceptable. But we should still seek to speed that up further. So moving on to slide 4, I think there actually are a number of partners involved in developing ATMPs in Europe and the US. And the virtuous circle on the left-hand side, I think, sums those up. As clinicians and clinical scientists, we want to innovate new treatments, and for that we need patients to be involved and participate. Universities for whom we work need high-impact publications, and that probably drives most of what we do. But the university also believes it wants intellectual property, but it's not in a position to fund phase 3 trials or indeed take these products through to a marketing authorization, which is where the real income can be generated. Industry, in the form of biopharma and pharmaceutical companies, needs clinicians and clinician scientists to innovate because these developments are still largely coming out of academic centres. And they need universities to support proof-of-principle studies funded mostly by academic funding sources. The university needs industry to buy its intellectual property, and after clinical trials it needs industry to commercialize that, and we'll talk about that later on in this presentation. But in the centre of all this, we have governments that need to ensure patient safety, but they also need to make available these novel therapies for patients because they have demands from patients for that. So patients need access to new, safe and effective treatments, and for that they need partnerships with clinicians to develop them and deliver them, and industry to manufacture them and supply them. And I think without that partnership, we can't deliver the numbers of doses that are required for a successful therapy. So if we move to slide 5, we have to ask ourselves how we're going to provide these advanced therapies to patients who need them, not just in our centre or in our city, but globally. And we have an option. We can stay in academia. Don't develop the product, just publish a paper, move on, and hope that somebody else takes it and runs with it. That has historically been the scenario for most of these academic developments, and I think it's actually unethical to put patients through high-risk procedures in high-risk early-phase clinical trials if your intention is not to develop it further. An alternative is a commercialized supply rather than a commercial supply, and I'll talk about that in a few slides' time. I'm also going to present some examples of academic spin-offs, companies that are created by academic developers and with the aim of delivering these therapies to patients. And I'm going to talk about spin-outs, where academics have got together with professional drug developers to develop a new company. And increasingly, as these companies exist and as large pharmaceutical companies get involved, there are examples I'm going to go through where these IP ideas and early phase data are sold to existing pharmaceutical or biopharmaceutical companies. So if we move to slide 6, I think all of those 4 options actually are predicated by a major question, which is how do you design the manufacturing process and a supply chain to allow you to source the starting material from a patient or sometimes a donor through a manufacturing process back to the patient for their therapy. And I think this is a major concern and a major weakness of a number of the products that I'm going to talk about, whether they be in academia, academic spin-out, spin-off, or even in large-scale pharmaceutical companies. Slide 7 starts to consider what one should look at early phase trials in order to guarantee success. And I think the first question to ask yourself as a drug developer or an academic developing these therapies is, what's your goal? Do you want a high-impact paper and then to walk away, or is your aim to change clinical practice by delivering a therapy that can be made available to the global population that needs it? In which case, you need to assess your global population. How many patients would actually need to be treated? A process that is typical for a phase 1 clinical trial in an academic setting might make 10, 20, maximally 30 doses, and it's unlikely to be scalable to treat 10,000 patients. A good example of this, I suppose, are tumour-infiltrating lymphocytes, which are one of the sort of stalwarts of cellular therapies going back many decades, and many patients have been treated successfully with TILs, particularly in melanoma. In order to make that a product that can treat the 10,000 patients that need these therapies, you will have to change the process, and even a therapeutic spin-out from NIH, where this therapy was invented, is struggling to work out how one would deliver the sort of doses required to meet a global need. It's very tempting to think that you could just robotize a manual open process to guarantee aseptic manufacture inside a mechanical system. That's actually very difficult, and I think it's unlikely to work. In the examples that we've tried, it doesn't work because a robot doesn't have all of the additional skills that an operator has that you don't necessarily realize that you're using. One way around it is to start closing the system down yourself and either using novel consumables that you can develop yourself or trying to find them off the shelf. And we've already started working with 3D printing prototypes, and we have an engineer in our team who's CAD competent, so that we can start thinking about how we might cheaply make a novel consumable that's specific to our manufacturing process. And once you have a 3D prototype that you can try, you can then get it manufactured. You also have to consider how you're going to make a supply chain that's secure to make 10,000 doses a year, with multiple suppliers of critical reagents and equipment. There's nothing more catastrophic in a manufacturing process, even for a clinical trial, let alone for a marketed product, if you only have one supplier of one critical piece of equipment and that supplier ceases to provide or can't support that piece of equipment. And there are plenty of examples of that already in the field. You also have to think about the logistics of your therapy. A typical phase 1 product is made very close to the patient, and my experience is very much that. We make our products in a GMP facility in a hospital for patients in our hospital or patients close by. When we did our first phase 3 multi-center trial, actually moving the product from our site to the end user wasn't so much of the problem. The greatest difficulty was actually bringing the patient's starting material from the remote site into our site in a way that the cells were viable. So this hub and spoke manufacture, where the patients are procured, or the material from the patients is procured in one hospital and then sent to a manufacturing site for the ATMP manufacture, is challenging because some of the starting materials actually have a very short shelf life. A good example of that is the production of monocyte-derived dendritic cells from peripheral blood samples from patients with a variety of cancers, in that those monocytes are very difficult to transduce into DCs over a protracted period if they're more than 36 or 48 hours old. So your starting material supply chain needs to be worked out beforehand, and if you've only treated patients who are 2 floors above you in your institution, you won't be aware that that supply chain is critical. Also, the stability of the final product in the supply chain is very important. A good example there are expanded NK cells, which have been expanded on the genetically modified K562 cell line from St. Jude's. Those cells are very, very potent anti-tumor NK cells, but they can't be shipped except in live culture. And when they arrive at the end user, they need to be washed, dosed, and then injected into the patient. And those are, in many ways, or in many legislations, considered manufacturing steps. And most hospitals don't have a license to undertake a manufacturing step of an ATMP. A further consideration is are some of the tumour cell therapies that are already being tested at early phase clinical trials, where the tumour cells must be irradiated prior to delivery. And obviously irradiation is a manufacturing step, and if that can't be done by the manufacturing site, it's very difficult to envisage a situation where that could be done by an end user site. A very important consideration, even in the early phase of clinical trial development, is how much is your therapy likely to be worth if it's successful. If it can replace an existing treatment, then plainly there is a known amount of money that is spent on those patients for that particular therapy, and therefore you can target that. A challenging example is the gene-modified stem cell therapies that are being used to cure single gene defects. And without mentioning companies that are involved in the field specifically, I'm sure many of you are aware that one can take autologous stem cells from an apheresis, select them using CD34 immunomagnetic selection, and then gene transduce them. Those patients require, therefore, an autologous stem cell transplant. Yet, the availability of autologous stem cell transplants globally is normally determined by the number of patients with the particular malignant condition that that stem cell transplant is used for example, lymphoma. It's very difficult to imagine a healthcare scenario where you can go to your hospital and say, your hospital is funded for 35 autologous stem cell transplants for lymphoma. And, oh, by the way, can I use 2 of them to treat patients with beta thalassemia, for example? So these challenges have to be worked out up front. The cost of your therapy is now not just the cost of making it, it's the cost of an autologous stem cell transplant, because the hospitals, the healthcare providers, is not going to suddenly be able to find the funding for additional autologous stem cell transplants. And it's this sort of understanding of the delivery situation in which your therapy is going to be used that allows you to appropriately cost your therapy and the delivery of that therapy. In terms of working out what it's worth, there is certainly a move within Europe to think about lifetime cost. And therefore, the value of your product can be assessed against not just the number of years of additional life it might give a patient, but the savings in other treatments that might be required for a patient with a chronic disease over their entire lifetime. And these health economic questions are things that really need to be looked at very early on in your trial considerations, because you might realize very early on that your therapy, although potentially very interesting, actually could never be delivered in an affordable healthcare environment. Moving on from that, how do you look at the cost of goods, and how do you make those cost of goods as low and as controllable as possible? And I think that one of the largest issues around cost of goods is how you manufacture it. Are you going to be manufacturing an aseptically produced product in a grade A environment, in a grade B background in Europe, or in a grade C background in the US? And we'll just think about some of the numbers there. A grade B lab costs approximately £2,500 a day in an academic centre to run, almost certainly more than that in a commercial centre. And if we think of CAR T cells as one of the sort of sexy ATMP therapies out there at the moment, a typical CAR T takes between 2 and 3 weeks to manufacture. So you're looking then just in the manufacturing costs of £35,000 to use the laboratory before you've bought any reagents or employed any staff. One of the solutions that people have been thinking about to CAR T manufacture is to move them into the semi-automated Miltenyi Prodigy device and stick that into a grade C lab. That halves your lab costs per day, but it requires £120,000 of capital expenditure for a 14-day product. That's a maximum of 25 products per year per device, and therefore to produce 10,000 CAR T doses a year, you need 400 Prodigy devices, which is £50 million in capital investment before you've even built your lab, paid the running cost of £1,200 per day, and employed the staff. But against that, 10,000 sales is a revenue of £1 billion if you can charge £100,000 per treatment, which gets back to the question at the top of the slide. So one of the challenges that we face throughout all of our early phase trials is, could we design a cheaper alternative? Can we semi-automate it? Can we close it? And could we run multiple production lines in a single grade C laboratory? And all of these are questions that one can ask at the academic stage of your trials, and not wait until they're commercialized. So if we look at slide 9, going back to how we provide these to patients who need them, we can stay in academia with a commercialized supply. By that I mean we don't seek a marketing authorization and make a licensed medicine like conventional medicines that you're familiar with buying across the counter or being prescribed. We supply them as an unlicensed medicine, and I'll go into the detail in a minute about how that might be done. But with an unlicensed medicine supplies in Europe and elsewhere in the world, you are actually reimbursed for the manufacture of the product, not for the product itself. And you can either supply that completely academically or within a healthcare scenario at cost of goods, or you can supply it for profit with a commercial partnership or even a commercial manufacturing organization. And I think some of the more esoteric or bespoke therapies, this is the way that they're going to be provided. I suppose a good example of that is in our next slide, which is some of the work that we've been doing in making decellularized larynx in a GMP-compliant process to treat patients with laryngeal carcinoma. And if you look at that slide on the top left-hand side, we have examples there of 4 different cadaveric donor larynxes that we've had from organ donors that we then decellularize. And initially, if you look at the image in the center here, this is a piece of trachea actually, but a similar process is used. This is a completely open process in a Ricordi chamber in a grade A and a grade B environment, decellularizing the donor larynx to remove donor cells so that we can then reconstitute it with that patient's own stem cells. Very early on, we realized that this was not feasible even to treat the number of patients that we plan to treat in our center alone, let alone the global need of 200 or 300 patients per year across Europe. And if you look at the image on the right-hand side of that, you can see that we've already moved this into semi-disposable bioreactor cassettes driven by a semi-automated device that pumps through the decellularization reagents. And in the bottom of the slide, you can see that being transferred directly into a GMP suite, within the Centre for Cell, Gene and Tissue Therapy at my unit, and completely closed, automated process for producing a decellularized trachea, decellularized larynx, and indeed in that image there, it's decellularizing an esophagus. So these are not hugely expensive devices to develop. They're not terribly complicated to develop. They just require a bit of thought up front. So moving on to slide 11, this is the schema for producing new ATMPs into a deliverable drug for patient need. So we start off with a new ATMP, and then we have a first-in-man application, which is almost, as I said from my very first slide, 90% of these are entirely academic. And that leads then to a phase 1 or phase 2 clinical trial, which is predominantly academic, but in some cases now these are being driven by industry. Because at that stage one can decide whether that product, if it works, could be commercialized and go through a traditional commercialization route to marketing authorization, or perhaps could never meet the standards of marketing authorization, in which case you could keep that manufacture still to GMP, but within an academic or a hospital setting. Or you could farm it out to a contract manufacturer, where the contract manufacturer gets paid for the manufacture, but not for the final product that's provided. And that actually can be done under UK special legislation, and increasingly the hospital exemption clause in Europe can be used to produce these cells in a hospital or academic setting. But now there's one example in a contract manufacturing setting. If one goes back to the phase 1, phase 2 trial option, if one decides at that stage that not only is this likely to be of commercial interest, but it could be commercialized, i.e. The manufacturing process could be tied down to meet marketing authorization standards, then it can go through the typical phase 2, phase 3 trials routes, increasingly moving from academia into industry, and then going through a drug development process through to marketing authorization and full industrialization. Even then, your industrialized product might still need some near-patient end-stage production, either within a GMP unit with a manufacturing authorization or within a hospital pharmacy. But all of these are considerations that need to be thought out in advance so that they can be tested during your phase 1 and phase 2 trials. So moving on to slide 12, an alternative to keeping it entirely academically is to create an academic spin-off, a new company which is led by the academic developers. And I won't embarrass people by naming companies that have had problems, but you can imagine that there are many of them that have faced the challenges listed here. Obviously, a very significant challenge is finding funding, and academics are actually not very good at going out and talking to commercial funding organizations, in my experience, largely because they're seen as not having the appropriate business acumen by venture capitalists or other commercial funding agencies. The academics also tend to have unrealistic or naive expectations of what investors will want to see, and without being too dismissive, I think there are very few examples of academic spin-offs that have been successful, certainly in Europe to date. However, an alternative is the academic spin-out. This is where you combine the knowledge and skill set of the academics who've designed the product, had the original idea, and maybe have done the original early-stage clinical trial, and then get into partnership with either a new company or an existing company to develop their product. And the examples here are companies which I've been involved in. Cellmedica was created and spun out some of our early-stage antiviral T cells. Fortress was a US company that took my NK therapy, and Immune Bio is doing the same with my second NK therapy. Now, having been involved in these spin-out companies, the challenges are somewhat the same. Funding is always a challenge. But the unrealistic and naive expectations now come in two ways. It's not just the academic having unrealistic or naive expectations, but quite often the industrial partner has an unrealistic expectation of what the academic can contribute, and naive expectations about how quickly these products can get through a clinical trial process. The complexity of the manufacturing process and the complexity of the product itself. However, companies quite often bring manufacturing expertise, they bring potential better capital investment, and staffing is generally easier to scale up with a commercial academic spin-out. One also has the option of partnering with a commercial manufacturing organisation, and we've successfully done that, and I'll allude to that later on. So a good example of this is the Holostem Advanced Therapies from the University of Modena, and their a product which recently got marketing authorisation, HoloClass. The final option is to sell to an existing pharmaceutical or biopharmaceutical company, and now that Cellmedica has been around for a few years, it already is doing this. It has a relationship with University College London and Baylor in Houston in Texas, where developments by academic scientists within those two institutions are proposed to Cellmedica to see if they want to buy them in. I suppose the most famous is probably Novartis' association with UPenn over the CAR-T product, and many of you will be aware that Pfizer formed a partnership with UCL over a human embryonic stem cell product for treatment of macular dystrophy. But I think if one looks at slide 15, the problem in 2014 and before was a funding problem. Once you'd finished your phase 1 or phase 2 trial at academia, you were sitting one side of a huge gap trying to find funding, either yourself or through those pathways that I described just now, to get enough money to do a phase 3 trial and ultimately to obtain marketing authorization. But funding is less of a problem at the moment, partly because of the success of CAR-T cells and the enthusiasm for such cell therapies, but also a greater interest in the whole concept of using patients' autologous cells to treat cancer or single gene defects. If you go to the next slide, slide 16, I think the problem now, last year and pressing on through 2017, is a technology gap. So one gets to the end of phase 1 or phase 2 with a product that's been made in an academic centre for a small number of patients, and now you're faced with a technology gap to make that product, either that process good enough to make product for phase 3, and once tied down then, the process that you take through to marketing authorization. And you get to a question, will the phase 3 trial ever enroll, because there's been such a delay in getting across that technology gap. So if we go to the next slide, I think this identifies the problem quite elegantly. You finished your phase 1, phase 2 trial in 10 or 20 patients, and you have a process that makes that product reliably, albeit not terribly efficiently. Now you need to go into your phase 3 trial, and I'm fully aware of companies that have not changed the process from phase 1 or phase 2 simply because they're terrified that by changing the process they may well change their product, and they don't want a phase 3 failure when they've had excellent phase 1, phase 2 data. The problem there is that you then have a process that's tied down to your marketing, when you get to your marketing authorization, that's completely incapable of delivering the number of doses that you need to deliver. So if we look at slide 18, I guess, why is this a problem? What is the difficulty in taking a product from phase 1 right the way through to marketing authorization? Well, if we look at slide 19, the phase 3 process, the process that you use to make your product in your phase 3 trial, is the process and the product that you're going to take through to your marketing authorization. At the end of phase 3, you can't make changes. So is that process fit for the thousands of doses that you're going to need to deliver commercially? And in many cases, I would say the majority of cases, that's not the case. Even packaging. I'm aware of a phase 3 trial, a commercial phase 3 trial, that was sending out end-user product in Nunc cryovials, a research-grade cryovial that had to be unscrewed. Now, if one just thinks about the most simple aspects of pharmaceutical vigilance in manufacturing, free particulates of unknown origin are one of the things that pharmacists get particularly anxious about. And yet the use of a polypropylene cryovial to deliver an end-stage product for injection into a patient, as soon as you scratch the side of that vial with your hypodermic needle, you're creating, by definition, particulates that are of unknown origin, i.e. scraped from the side of your cryovial, and injecting those into a patient. And yet large commercial companies have done clinical trials at phase 3 using cryovials to deliver their products. Labelling. I quite often see problems where the size of the packaging doesn't meet the labelling requirements for a marketing authorization product. So this needs to be considered right up front. What is your label going to look like? What are you going to be required to do? And then supply chain. Nearly all cryopreserved or frozen cell therapies are going out still in nitrogen vapor in dry shippers. And you have to think, how large a facility do you need to produce the number of dry shippers that you need to supply a 10,000 product a year patient demand? So let's look at slide 20. Here is a photograph of a newly available GMP-compliant device for filling and closing NUNC cryovials or similar research-grade cryovials in a GMP-compliant process. And yet, to use a cryovial, even in the manufacturing process, let alone as the final delivery product, is a failure because it forces you to use an open process in manufacture. It's time to dump cryovials, and we should be already using the variety of alternatives that are out there. So let's look at the problems of this phase 2 to phase 3 technology gap in a bit of detail. And I won't use specific company names, but I'm sure you can think of them. So a biopharma company or a large pharmaceutical company buys a new ATMP or buys the intellectual property for an ATMP from University Y. University Y has already conducted a very good, successful phase 1, phase 2 trial in a small number of patients with an incurable cancer. And so the company then runs a small phase 2 trial at the same university, and they get the same great results. So they now decide to expand the phase 2, and in that immediately the company realizes that the supply chain, the manufacturing process, and the cost of goods is just not compatible with the commercial market size that they're going to have to deliver to if this phase 3 trial works. So now they have to sit down and work out how they're going to make the product for the phase 3 trial. And that requires many years of process development. I've put 3 years here, but it can be much more than that. Assay development, assays which are qualified and ultimately validated to show that the product that you made in your phase 1, phase 2 process is the same as the product you're making in your phase 3. And maybe even a bridging study to show clinical compatibility between the product made in the phase 1 and phase 3 process. So now, 3 or 4 years after you finished your terribly exciting phase 2, you're ready to complete your phase 3. But by now, you have a generation 1 or generation 2 product, and the academic investigators have moved on to the next generation or even several iterations further on. And as an academic investigator interested in a high-impact publication, they're not particularly interested in enrolling patients into your phase 3 trial of a first-generation product. So the trial never gets done, or at least is very difficult to get done, and delays even further. If one had solved that technology gap at phase 1, phase 2, one certainly wouldn't have had the 3 years of process development, and one could have gone straight from phase 2 into phase 3 and marketing authorization. So if we look at how one might do that, the challenges involved in getting ready for phase 3 are closing the process. I think the open processes for a marketing authorization product is going to be very, very challenging. And for that, we need equipment, we need compatible plastics, and we need to show comparability in terms of the quality of the product with the old open process that almost certainly would have been the starting point. We need good QC assays, quality control assays, and for that we need the appropriate reagents, equipment, controls, and a quality assurance system to show that the assays that we're applying actually are reproducible. And we do need informative potency assays, and the best way of obtaining those potency assays are these, that's called for information-only data from early phase trials, where one runs a number of different potency assays and tries to then retrospectively show if any of these have been associated with clinical efficacy, and therefore can be validated as a potency assay. The other critical thing is redundancy. as I said earlier on, the most important thing in the manufacture of these products is to make certain that every part of that chain, whether it be a reagent or a piece of equipment, is available from more than one supplier, so that you don't lose your entire product just because one supplier goes out of business or no longer supplies your critical reagent or equipment. And finally, the cold supply chain. I think if we can validate the shipping of our products on dry ice rather than in nitrogen vapor, we really are assisting the downstream deliverability of our product when it gets a marketing authorization. So just to give an example of how we address this with our very first cell therapy, back in the late 1990s, early 2000s, I discovered a way of priming human resting natural killer cells to make them target NK-resistant tumors. And we used a tumor cell line called CTV1, to provide that priming signal. So we developed a phase 1 GMP process, we sourced our cells, we worked out how to expand them, and we worked out very early on that delivering a cryopreserved cell itself was actually going to be challenging, and we had a lot of work to generate a lysate of those cells, and showing that that lysate not only was stable at storage temperatures of -80, but that lysate could be reproducibly manufactured at scale. We identified a clinical grade NK cell selection process, initially from one supplier, but then from a second, and we proved that removed the T cells to what we believed would be adequate for the clinical trial, but obviously until one's completed the clinical trial, I couldn't say it was adequate. And we then looked at the effect of that T cell depletion on the trial design. Did it determine a maximum dose of NK cells that you could achieve having removed so many T cells? We then engineered the phase 1 GMP process. We scaled up and we set assays for the lysate to demonstrate comparability between batches. And then we validated the phase 1 and conducted a successful phase 1 academically in a single centre in London. The data were interesting, and the IP was sold to a US biopharma company, and we then had to transfer that technology into the US. And I won't labour the point, you can read the slide. We used two different contract manufacturers in the US for the phase 1 trial, and in order to do that, we had to be able to show comparability of the product made in London, made at CMO1, and made at CMO2. And for that, we needed to develop, as highlighted in red here, the assays to prove that the product was the same irrespective of where it was made. Now, this is a common problem for companies developing ATMPs, and if one just goes to slide 25, without labouring the point here, this is academic preclinical work that we did back in the early 2000s, showing that the CTV1 membrane, when it binds to the NK cell, it sends a number of signals to the NK cell, but one of the critical ones is through the CD2 molecule on the NK cell. And what we mapped, and I say I won't labour the point, these data are published, what we mapped was the entire signal transduction pathway in the NK cell. And by knowing that degree of depth of how the CTV1 interacted with the NK cell and how it created this tumor primed model, we were able then to use that same intracellular signaling pathway to guarantee that the cells manufactured in other centres had the same downstream signaling, and ultimately in our functional potency assay delivered the same cell. If we go into slide 26, what that then did was allow us to semi-automate the process. On the left-hand side, you can see our original process using a conventional Clinimax for CD56 selection and CD3 depletion, and then initially a large-scale tissue culture flask of T175 to generate the product, followed by a manual density gradient isolation step to remove the lysate, and then finally packaging in a cryobag to be delivered to the patient. Transferring that all onto the semi-automated Clinimax prodigy allowed us not only to close the system down entirely, to move it out of a grade A and a B background into a grade C lab, but it also increased our yield. And so whereas we'd only produced one bag of product or one dose of product in most cases in the old manual system, we now got between 4 and 6 doses of product. out of a closed, reproducible, semi-automated system. So there are huge benefits to be made in scaling out, but one has to think of what the limitations are during that design process. So moving to slide 27, what we ended up with the question of how do you validate this significant process change? We'd gone from a manual process to a semi-automated closed process, and did it make the same tumor-primed NK cells? Well, once again, we went back to our detailed knowledge of the product. We knew the characteristics of the timing preparation, we knew the signaling pathway, and we had qualified potency assays. And by taking all of those to the FDA, we were able to convince them that we had a manufacturing process that produced the same product. So this is where academics, the academic knowledge of the product and the process actually enhances the ability of the company to develop the process, because you can bring in this detailed knowledge of your product. If we go to slide 28, the other issue that we have to consider is scale. This is an image of a US contract manufacturer producing mesenchymal stromal cells, or mesenchymal stromal stem cells, in conventional cell stacks. And this is a good example of where a research-grade system making a product for a small number of patients basically has just been extended. So no great thought, no great invention here, just a much larger version of a preclinical or early clinical system, with considerable problems in terms of fluid handling and very little control of cost of goods. If you look at the next slide, the same number of cells can be produced in disposable bioreactors. This is the PAL-ATME expansion system. We now have this routinely in use in our laboratory, making clinical grade MSCs in a semi-automated but entirely closed manner. Now this has been challenging to set up, but ultimately it allows us to produce between 2 and 4 times 10 to the 9 MSCs in one production run, which is comparable in one production run of a 23-litre bioreactor, which is comparable to the number of MSCs that were being derived from the large-scale manual system in the previous slide. So why would a small academic unit like ourselves get involved in such large-scale manufacture of MSCs? If you go to slide 30, you can see an example here of what we're using that for. This is a trial called Tactical, and it's generating a large cell bank of allogeneic MSCs from normal healthy donors that are transduced to express TRAIL, the TNF receptor apoptosis-inducing ligand. And TRAIL obviously is a molecule used by T cells and NK cells to target and kill tumor cells. So we believe, and though we have preclinical data to support the fact that MSCs carrying TRAIL can track to the site of a tumor and induce tumor death through TRAIL signaling. So in order to manufacture that product for a clinical trial in 30 patients at an academic phase 1, phase 2 level, we've taken multiple donor bone marrow or cord, and then isolated MSCs from that in an open process, as you can see here in T175 flasks. And then that small number of cells at the end of that first passage, we transduce with a lentiviral vector to express TRAIL, and then transfer these into a master cell bank, which is cryopreserved, and a working cell bank, which goes into one of these large-scale bioreactors. You can see that at the end of this, we're looking at producing between 5 times 10 to the 10 and 3 times 10 to the 11 transduced MSCs, not just for the phase 1 trial, but if the phase 1, phase 2 trial is successful, that cell bank is then a validated cell bank which is already manufactured and can be used to go straight into phase 3, and it actually provides a large enough cell bank to obtain marketing authorization before having to be remanufactured. The other consideration here is that by using multiple donors to create our master cell bank, we actually have the value of a heterogeneous product at outset, and we're not simply using a single donor and having to say that when we manufacture from another single donor, we would have the same degree of comparability. So I've given some examples of academic trials in which we've considered from the very beginning how you deliver these products to the number of patients that are ultimately going to need to be supported. So in conclusion, I think it's never too early to consider how you close a process to control both manufacturing cost of goods, but also enhance deliverability. I think it's actually quite easy now to develop phase 1 processes that actually can be fit for the marketing authorized product. And that gives you added value when you're looking for your buyer or your investor in your intellectual property after you've finished your phase 1, because you can show to them that they've got a seamless walk through phase 1, phase 2, phase 3, and marketing authorization application. But academics are experts at studying basic biology, and I think that's really the other added value that we give here. The more we know about our product, the easier it is to facilitate comparability studies and to manage the change process. It also gives us the opportunity, because of our academic knowledge, to develop really good QC assays during the early phases, which can be used not just for control of product changes, but also further on in the manufacture of the product for marketing authorization. But finally, I think the most important message I want to give is that a delay between a successful phase 2 and the start of the phase 3 really can be the gap of doom that kills off many of these therapies. Thank you very much.
Thank you very much, Mark. Wonderful presentation. We'll now move on to answer some of the questions that have come in from the listeners. The first question we have is, do you think the draft plans from the EMA for GMP Light for ATMPs will have a positive or negative impact on the field?
Thanks very much. This is quite interesting and quite controversial. So for those of you that aren't aware, there was a discussion document put out by the EMA late last year with the aim of asking the interested parties around Europe whether there was any enthusiasm for reducing the demands for GMP compliance for the manufacture of ATMPs, particularly in the academic setting. Now, I think that obviously this has the positive benefit of being able to increase the availability of early stage ATMPs into academic clinical trial. The thing that concerns me, and I think it concerns a lot of the field, including PICS, is that by doing that we end up with two standards of GMP, and products that are made to that standard in Europe wouldn't be recognised for trials outside of Europe. That's a retrograde step, considering the international nature of the development of these products. And I think it also risks the potential of making products that get into early phase clinical trials that the process of manufacture is just not fit for commercialization later on, and so this valley of doom of technology gap actually worsens. So I'm not in favour of it, but there are positive benefits in terms of it getting potentially more products through early phase clinical trials.
Fantastic. Thank you, Mark. Just a note to everyone, this is an opportunity to ask any more questions, because at the moment that's the only one we have. So if you have something you want to ask Mark, then please ask it now. Otherwise, I suppose we'll have to wrap the session up. So we'll give you a few minutes to send anything through. Oh, well, Mark, you must have addressed everyone's questions in your presentation, because no one seems to be asking anything today. So I guess with that, we'll wrap the session up, and again, say thank you very much to Mark for his presentation. Just to make you all aware, we do have another session starting in roughly 11 minutes with Janneke Mullenberg, who's the CEO of Vicinivax, and she's going to be speaking on clinical development of VVAX 001, which is a semi-key virus-based cancer vaccine for the treatment of HPV-related pre-malignancies. And yes, as I said, that will be starting again in 11 minutes. So thank you ever so much, Mark. And to join it, you'll need to exit this screen, go to the main lobby, and then click through to the new session. Thank you.